Great. Good morning, everyone. I'd like to thank the organizers for inviting me today to talk about DNA methylation biomarkers of environmental exposures and to think about how we might use these as a tool for cancer investigations. Here are my disclosures. And I wanted to start, since it's a broad audience, just defining what DNA methylation is, since that'll be the focus of this talk. DNA methylation is the addition of a methyl group on cytosine nucleotides in our DNA sequence, so the set of A's, C's, G's, and T's. This primarily occurs at what we call CpGs in humans, or C's followed by G's. There are about 28 million of these in the human genome, and they play a key role in many cell processes and functions. They can be added or removed without a change in the DNA sequence itself, and these patterns are remembered when a cell divides. There's growing evidence that DNA methylation is susceptible to environmental exposures in humans. The evidence has come both from observational epidemiology literature, as well as lab toxicology studies, and a wide range of different environmental exposure domains have been identified, including toxicants, nutrients, pharmaceuticals, and even lifestyle and demographic factors. So why are these methylation changes associated with exposures important? Well, the first reason is they could inform our understanding of disease mechanisms. So they could be part of the cause of a health outcome. The second way they can be useful is they might serve as a biomarker of an exposure. So in this example, they're not part of the causal pathway between an exposure and a health outcome, but they're simply a marker of an exposure. And then the third way they can be important in our understanding is serving as a biomarker for health outcomes or disease. So they could either be useful for predicting future disease or outcomes, or they could be a consequence of the disease, which is also really important and can provide a marker of disease. And so today we're really just going to focus on the utility of methylation as a biomarker of exposure, with a particular focus on cancer investigations. So what have we learned so far about methylation biomarkers of exposure? I'm going to tell you a story and use evidence from smoking. And that's not because I think that's a difficult one for us to measure, or one that we are trying to identify for cancer risk. That's pretty well known. I think it's because it really provides proof of principle evidence for how this kind of methylation tool could be useful for other exposures we might be interested in examining for cancer risk. So what have we learned for smoking? The first thing we learned is that DNA methylation patterns can reflect environmental exposures, including prenatal exposures. So prenatal exposure to smoking has been associated with differences in methylation patterns in birth samples. Here's an example of just what a pattern looks like at 26 CpGs related to prenatal smoking exposure. DNA methylation patterns have also been detectable in childhood blood samples, and they reflect their past historic prenatal exposure. And then furthermore, these patterns now at age 5, which are strikingly similar to those that were discovered at birth, can predict prenatal smoking exposure in 5-year-old children with 87% accuracy. The patterns of methylation related to prenatal smoking have also been discovered and are sustained into adolescence. And this is even after accounting for postnatal exposure. So it appears to be independent of postnatal exposures. Here's an example of just one CpG where the difference between the red and the blue lines, which is prenatal exposed versus non-exposed, persists from birth up to age 18. And then finally, evidence has shown in 30 to 53-year-old adults, the prenatal changes in methylation are also detectable. And importantly, these are independent of personal smoking history. And so that was evidence for smoking, showing across the lifespan that there are methylation patterns and changes that are detectable in blood that are specific to prenatal exposure. There's other evidence for other exposures. I'm just showing an example here for childhood adversity, where most of the methylation patterns at age 7 related to adversity are related to, again, specific sensitive periods of exposure in very early childhood. But there was also some evidence from this study showing that methylation patterns might also capture cumulative levels of adversity exposure or recent adversity exposures. And then finally, there are many other classifiers that are being developed. So there's a great methylation classifier for self-smoking history. And then there are many others as well being developed for other exposures. And so another question we wanted to ask, and what we've learned about methylation biomarkers for smoking, is we wanted to know, can you use the methylation values themselves as a measure of exposure to detect exposures associated with cancer risk? And the sort of perfect example here for us is, can we use the smoking methylation score in adults to predict their incident or future cancer risk? And when we did this in ERIC, we found that for lung and aerodigestive cancers, participants had a significant increase in the hazard risk for incident cancer if they had the highest methylation smoking scores compared to the lowest. But we did not see an association between the methylation score for smoking and prostate or breast cancer risk, which is what we expected, since we know those two types of cancers are not related to smoking history. And so this sort of checked our box for showing that, yes, you can use methylation values themselves, sort of in lieu of more traditional exposure collection methods and metrics, to detect and identify exposures that are risk factors for cancer. And then furthermore, another interesting thing we found in the study is, if we looked at, again, the risk of cancer using a methylation smoking score, and you adjust for self-reported pack years, again, collected through more traditional ascertainment methods, we still found for lung and aerodigestive cancers a significant increase in risk of those cancers. And what this indicated to us is that methylation itself appears to carry an additional or kind of residual risk information that we missed by just using self-reported pack years. Another interesting sort of observation we had in this study, which again has really important, I think, food for thought implications for what we're going to be discussing today and thinking about clusters of individuals, is when we compared the smoking methylation scores, which is shown on the y-axis here, with the higher indicating they had more smoking history or a greater smoking history, lower values here indicate less smoking or no smoking. And we compared that to their self-reported pack years. So again, the highest smokers were on the right, lowest smoke years were on the left. We observed this cluster of individuals that had extremely high methylation smoking scores, but very low or even no smoking history. And so we dug a little bit further to try and figure out and think about what might be going on with these participants. And so there are a couple of possibilities. The first is, maybe these participants had less robust data collection. That's possible. It might be that they misreported exposure, right? So they said they were non-smokers and never smoked in their life, but actually maybe they did. And then there might actually be something else, which really intrigued us when we did some digging and figured out all of these participants came from the Forsyth County, North Carolina site, which is a very large tobacco and cigarette production area. And so it makes us wonder, although we don't know and we can't prove which of these is going on to explain this cluster, but might they have actually had exposure to tobacco burns when they were clearing the lands? Or might they be employed in tobacco production factories? And so I've given you now a lot of lines of indication for how methylation patterns that reflect or biomarkers of exposures might help us to overcome challenges with current study designs and open new possibilities, and how it might provide a complementary measure of exposure, again, to help us when we're thinking about discovering risk factors for cancer. And then there are lots of future lines of research we need, I think, to realize the full potential. I don't have time to go into each one of these right now, but we can discuss them later today. And then finally, I wanted to share with you two sort of big and bold thoughts I had. So it's a bit provocative, but I think it makes us to start thinking about what else we might be able to do with methylation biomarkers in cancer investigations. And the first idea I wanted to throw out there is, can we use methylation, let's say, as a biodetector or a way to detect shared exposures among cancer clusters or cases? And so here's how that might look. We would collect methylation patterns from a sample of individuals. Let's say we have a population of individuals with cancer and without cancer that we're investigating. We could then take those methylation patterns and we could compare them to a database that contains the patterns for different exposures. And then using those, we can deconvolve and figure out, are there specific exposure methylation patterns that are more common in individuals with cancer? So I'm going to call those cancer-associated exposure profiles compared to the individuals that didn't have cancer. And so this could be a way for us to think about and prioritize exposures to study to see if they are related to cancer in those individuals with cancer. This might be improved by combining this epigenetic biomarkers of exposure with genetic variant patterns. And then we can also consider designing custom arrays to improve the cost efficiency of doing these types of studies, where we focus on exposure methylation measures. And then finally, the last sort of big and bold idea I wanted to throw out there for us to sort of go through the thought process on is, can we use methylation biodetectors to be able to monitor cancer risk in populations? And this could be really informative because it can help advance precision public health and precision medicine efforts, right? So it can help us to identify possible high-risk subpopulations or individuals that we could add additional screening recommendations for, and that can help us prioritize our limited, usually intervention resources. So again, how might this look? So we would take patterns from a population of individuals, methylation patterns. We could then compare those to our reference exposure biomarker database and cluster the methylation patterns of individuals to identify groups that fit a low risk exposure profile. Let's say there might be a subset of individuals that have a profile that suggests they have a high lead exposure or they fit in a high lead exposure group. And maybe there's, other groups for NO2 and many other exposures. And once we then group them by their methylation exposure profiles, we can then use that information, let's say, to output recommendations for particular subgroups, right, or individuals. And that could be additional screening at the individual level or even at a subpopulation level. And then it can also help us to focus interventions should some of these groups correspond to geographic or maybe demographic or socio-demographic types of subgroups in the population. And then finally, there may be groups where they're at low risk. They have a low exposure profile risk, and so their population would have no further recommendations. And again, I think it's important to highlight that this could be improved by combining with genetic variant and other omics kinds of biomarkers. And then by considering custom arrays, we can improve the cost efficiency of this type of surveillance and monitoring. So I'd like to thank you all, and I look forward to discussion.